So now let us look what is happening inside this neuron cell, which makes the neuron electrically excitable. There is also muscle cells are also electrically excitable. You should understand. Neurons and muscle cells are electrically excitable. So what make them special to be excited electrically is, to understand this, we need to go inside the cell. How charge difference or a polarity is being created inside the cell, we need to study right now. So let us go and study sodium potassium pump first, and also certain associated channels which makes the cell in a electrically excitable or a cell having a potential gradient within it. Okay. So before we go towards the pump, we should understand something about the gradients or concentration gradient or electrochemical gradient or electrochemical gradient, these things in a cell. I am drawing a plasma. Look, consider I'm drawing a cell here. Okay, look, a cell will be have this is a cell. Consider it as a cell. Usually it is having a plasma membrane within it, right? Plasma membrane. This is a plasma membrane. So cell is having an outer membrane of the plasma membrane and also an inner membrane, because we know that plasma membrane is a lipid bilayer. Okay, it consists of lipid bilayer. You just think of your house, which is gated and it is having wall, surrounding wall, which protects your house. So consider or relate it with the cell. So anything that should come into your house should enter through your gate, right? Or that is one method. The other method is the person should jump and cross your wall. Okay, just consider these two situations. So a cell is having a membrane which covers the cell, which protects the cell from exterior environment, which also maintains some internal environment inside the cell. So any molecule which is produced inside the cell or which should come inside the cell should cross the plasma membrane. You should understand, every molecules can't cross like this. Only few molecules can easily get through the membrane. It means it can move through the membrane directly. So if a molecule can move through the membrane, it should have the same character of the membrane, which is it has been made, right? It should be hydrophobic in nature, so it can easily pass through. You should also remember, all hydrophobic molecules can't easily pass through the cell. Only small hydrophobic molecules, because the cell is made up of lipid bilayer, which is hydrophobic in nature. So you should have a basic understanding about this thing. So certain molecules should cross the cell to come inside, and certain molecules produced inside the cell should go out. So this is maintained, or this is done with the help of certain protein molecules. What are actually protein molecules? We know that proteins are made up of amino acids. Thousands of amino acids get folded into primary structure, then secondary, tertiary, quaternary, then a protein will be formed. So in the plasma membrane of a cell, there will be presence of certain protein molecules. I am drawing it. It has in the shape of a box. This is protein molecule of one type, box. This is protein molecule of second type, circle. I'm just drawing it for your understanding. And certain molecules like phosphase. So a cell in its plasma membrane will be having a large variety types of protein molecules, which allow certain molecules to enter from the outside and also from inside to outside. This is the basic idea you should know. So if we are looking this diagram, just consider a membrane here. I'm just consider this as a plasma membrane, the line which I am drawing. There is actually three type of movement. Active movement and passive movement. Just consider this as active movement and passive movement. So what is an active movement? First we will go to passive movement. What is actually a passive movement? Look, I will brief you with a small example for an idea. At your home, we all use water for several purposes, right? So when we open the tap, water comes from the tank which is stored on the terrace or on the top floor of your building or your house. So water from the top will come down without any use of energy, right? We don't use any motor for opening the, we don't use any motor for taking the water down. It comes automatically as we open the tap, the barrier is removed and the water comes through the tap. It does not need any energy. Why it does not need any energy? Because there is an energy difference. The tank is being stored in a higher energy with a higher potential energy. So that energy is converted into kinetic energy, which is used by the water to flow down. And how the water in your tank gets there is by taking the water from your well, which is situated down from to a top height. So this process needs an energy. So this is an active process. But the latter or the first one which I have mentioned was a passive process because it does not need any energy. But the water from your well to your tank should be pumped. I am mentioning it as pumped. You should remember it is a pump. Pump means it needs energy. It is, we use a motor to pump the water from the well to your tank, which is against the energy gradients, right? The water is moving from low energy to high energy form, high height. So it needs the energy used by a pump. The same principle we can relate it inside a cell also. So water is being pumped from the well to your tank. From the tank, we use the energy to in energy stored in the form of potential energy to as a kinetic energy and used inside your house. So you should have a basic understanding about the idea. Now going to the cell, just consider the line drawn here is a plasma membrane. There are actually two processes, active diffusion and passive diffusion. Passive diffusion does not require any energy because the molecules is being moved from high concentration to low concentration, no energy required. So the first diagram and the second diagram is a example of, just consider first as an example of passive process because there is high concentration of blue dots, which means some solutes, any molecule, solutes, iron, anything. So in the right side there is high concentration of the solute, which in a membrane, if there is a pores in the membrane, it can easily cross the membrane and enter into a low concentration area. You should understand that this diffusion is only happening because the membrane is allowing it. If the membrane is very thick, it won't diffuse. So membrane also have a role here. Okay, so high concentration to low concentration, the molecule is being moved. That is passive process. No energy required. A downhill movement. Energy is being released, right? A downhill movement. Just consider you pushing a rock up a hill. It needs energy. But if you are coming down a hill, the rock will fall automatically, normally, without a cost of energy. This is the downhill movement. So simple diffusion is a downhill movement. Active diffusion is a uphill movement, which it needs energy. In the third diagram, you can see it. A molecule is being moved from low concentration area to high concentration area. Usually this is not energetically favorable. This is energetically unfavorable because a molecule is being moving from low to high. So it needs some energy. And you should also remember that there is a barrier in between these two concentration gradients. So barrier also should allow. So in case of active transport, there we use the pump. Pumps are being used there, which move. You should remember, active transport is the usage of energy. It needs some energy. So we use pump there. Pump means movement of a molecule against its concentration gradient, which means uphill movement from low concentration to high concentration. So this is active transport. Next is facilitated transport. Facilitated transport. A good example is the glucose in your cell. We will talk it first generally. Look, a molecule can move from high concentration to low concentration, but due to the presence of certain barrier, they can't cross. Due to the presence of, let's take an example of plasma membrane. It can't move to its low diffusion energy, because low diffusion, although it is energetically favorable, right, high to low, but it can't cross because of the high, let us consider high thickness of the barrier. So what happens here? Certain transporters come into the scene. What happens? Transporters take this molecule from high concentration to low concentration without the use of any energy. Okay, only this is to remember that only energy is being used in case of pump. Energy is utilized only in case of pump, because it is pumping something from low to high. So here you can see a red color round structure, which is a pump. Here it is a transporter. Everything is actually transporter, which is made up of protein molecules. Okay, proteins are made up of amino acids. Due to amino acids, these proteins work. There is actually a lot more to study in this, but for NEET purpose, you just should have a basic understanding. Actually, this much are not to be said, but I am saying for your purpose of understanding. Okay, so these three conditions are to be remembered. It is very important, because we are going to deal with in the next coming part. So active transport is done with the help of energy utilization. It is a pump. Pumps only use energy. Various type of pumps are present in a cell. Some of the examples of pump are P type, V type, F type, ABC. That is actually not needed. I am just saying for your understanding. P, V, F, ABC. The pump which we are going to study here is the sodium potassium pump, which is a type of P pump. So what is a P pump? P pump is a pump which needs phosphorylation for its activation. That is why it's given as P. P means phosphorylation. What is phosphorylation? Just addition of a phosphate group. Actually, it is phosphoric acid which gives the phosphate group. P, phosphate group, to the protein, which means the pump. Pump is a protein molecule, which is seen in a plasma membrane. So sodium potassium pump is a pump which uses the energy. It needs phosphorylation, P. So it is a class of P type pump. Just need to understand this thing. There are many other pumps also, B type pump, which makes an example is H plus ATPase. ATPase means utilization of ATP. ATPase is an enzyme, right? ATPase is an enzyme. It means it breaks the ATP. Ase in the suffix means it is an enzyme, active enzyme. So this enzyme, which ATPase, which means sodium potassium pump is an ATPase, it means that they have the ability to break the ATP. That is the reason they are given as ATPase. Okay. Now we are going to study sodium potassium pump. You should be listening it very much carefully, because it is very more important to understand. Very much important to understand. Okay, so I think you just got an understanding about three types of movement. Okay, now so let us go to sodium potassium pump. Just look the heading, sodium potassium pump, a pump which pumps sodium and potassium. That's the thing. We should understand from where to where sodium is transported and from where to where potassium is transported. Before that, let us consider a cell. This is a cell plasma membrane, lipid bilayer cell plasma membrane. You should have 2-3 points in your mind. Please note it down if you want. In a cell, usually cell in its extracellular site and intracellular site. Intracellular means in the cytosol or cytoplasm. Extracellular site means outside plasma membrane or outside the cell, that's outside the system. There are many ions present in our cell or body. So, and this ion concentration is different inside the cell and outside the cell. So here we are going to study about sodium potassium. I will say the concentration of sodium potassium outside and inside. Usually sodium is an extracellular ion. What? Sodium is extracellular ion. It means sodium is present in a high concentration extracellularly. And potassium is intracellular ion, which means potassium is present inside a cell in a high concentration. It does not mean potassium is seen outside. Potassium is seen outside, but compared with the concentration of sodium, potassium is very low. Okay. So actually it is 145 millimolar concentration of sodium is seen outside. It's actually 154 millimolar, and 10 millimolar concentration of sodium is seen, sodium is seen inside, and also vice versa. 150, 145 millimolar potassium seen in high concentration inside, and sodium like 10 millimolar outside. Just a general approximate value. So now you understood. Sodium is seen outside morally, extracellularly in high concentration, and in intracellular low concentration, but potassium is high inside. So what does the sodium potassium pump do? Sodium potassium pump does is that it pumps 3 sodium outside and 2 potassium inside. So sodium potassium pump will be present here somewhere in the plasma membrane. What it does do? It pump 3 sodium out and it pump 2 potassium inside. Actually, sodium and potassium are ions and they exist in their cationic stage plus. Okay, that is the condition. So what does this pump do? Pump does is efflux of 3 sodium outside and influx of 2 potassium inside. This is actually the working of pump. So if a pump work one time, we will be getting 3 outside and 2 inside. Okay, that is the thing. So now you will be getting something understanding, right? Because I said that pump is a protein molecule or a transporter which moves the molecule against its concentration gradient. Look, sodium is being transported from intra to extra. Sodium is a extracellular ion, right? So sodium is being transported from intra to extra towards high concentration. Same thing for potassium, from extra to intra. Sodium, potassium is seen high concentration inside, so it has been transported from low to high. So they require energy. This energy is provided by an ATP molecule, adenosine triphosphate, which means an adenosine and triphosphate, 3-phosphate group. What is an ATP? Adenosine triphosphate. It is an energy molecule. Why it is an energy molecule? We will see how it provides energy. How it provides energy? We always say ATP is an energy molecule. We eat food for getting energy, which is provided by ATP in the powerhouse of the cell, mitochondria, which makes the large amount of ATP there. We will be studying in chapter number 12. Okay, this is the condition of sodium potassium pump. So pump uses phosphorylation. Phosphorylation means addition of phosphate group. This phosphate group is given by, usually it is given by phosphoric acid. So let us see ATP molecule here. I am saying phosphorylation. There will be a little bit confusion because ATP, adenosine triphosphate, ATP is already having 3-phosphate. I am saying that phosphoric acid was the group which donated phosphate to the ATP. That is the thing I said. Okay, you should understand it. Not here. Phosphoric acid is not giving phosphate here. Here the phosphate is given by ATP, by the breaking of ATP. The adenosine and 3-phosphate, this is joined together and 1 phosphate is being breaked. So adenosine diphosphate. It will become adenosine diphosphate and 1 phosphate is released. That phosphate is used for the purpose of transporting sodium outside and potassium inside. So let us go through the diagram. It is a very easy process. I will just say in steps. So you can understand it. Look into the please look into the first part of the diagram. What happens? This is the step 1. Step 1. ATP will be binded. Please look into the diagram. ATP will be binded, ATP will be in the bounded form in pump, sodium potassium pump. This blue color is the sodium potassium pump. ATP will be already binded there. So this binding of the ATP will promote ATP will be already bounded here. So this ATP will be having ATP bounded pump will be having affinity for sodium ion. Okay, ATP bounded pump will be having affinity for sodium ion, and it can bind 3 sodium ion. Okay, when 3 sodium ion binds, it will make changes in the protein molecule, which when 3 sodium bind to the pump, it will make the ATP cleave. ATP cleave means it will be removing one phosphate from the ATP. So ATP will be become ADP and it will be removed from the cell, from the pump, and thus one phosphate will be attached to the pump. This is the first 2 steps. So why only 3 sodium is being attached? It is because the structure of pump is like that. I mentioned pump is a protein molecule. So this protein can only accommodate 3 sodium at a time. And when the sodium is being binded, the pump has an inherent ATPase activity, which means the pump is having ATP breaking capacity. So what does this pump do? When 3 sodium binds to the pump breaks the ATP, thus 2 ADP molecule, adenosine diphosphate molecule, is being removed, 1 phosphate remains. This phosphate, now we can say that the pump is in the phosphorylated condition, means the phosphate group has been added. So at this condition, when this phosphate group has been added, the pump will have a conformational change. Conformational change means it is a protein molecule. When there is a change in charge, the protein molecule can, this pump will make a change so that it will eject 3 sodium outside of the cell, which means to the extracellular site. This is how 3 sodium is being transported outside the cell. I think you got a basic idea. Okay, this much are not need to be studied. I am just saying it for your understanding. So 3 sodium has been transported inside, outside the cell in a phosphorylated condition. I will say once more, when the ATP is bounded with the pump, it has affinity for ion, sodium ion. So sodium ion binds. This binding promote the cleavage of the ATP by the pump, because the binding will promote the activity, activation of ATPase activity of the pump, which will cleave the ATP, forming ADP and a phosphorus group. In this phosphorylated condition, the pump will open and it will allow the removal of sodium ion outside. So in the third figure, it is saying that it's removing the sodium ion outside the cell, extracellularly. So in the phosphated condition, you should remember that in the phosphated condition, the pump will have high affinity towards potassium ion. So potassium ion bind from the extracellular. Extracellularly also there is potassium ion. So 2 potassium ion can bind extracellularly to the site of the pump. When this potassium bind, the phosphate group get released. When the phosphate group is released, it then have a conformational change and opens the channel or opens the pump towards the intracellular side. So at the intracellular side, 2 potassium is being released to the cytoplasm. This is how the transportation of sodium and 3 sodium and 2 potassium occur. It is completely based on the structure of the pump. There is no question why there is a transport only 3, not 6. It is because the pump can only accommodate 3 sodium at a time. So the resultant is the 3 efflux and 2 influx. So in a normal cell or a resting cell, which means the cell without any action potential, this is the condition. The potassium pump will be continuously working, which means it will be pumping continuously 3 sodium outside, 2 potassium inside, like it will be continuously pumping. So how does this create a result in a cell? That we will now understand. So this is the steps. If you want to write down the steps, you can write down the steps. You can pause the video, and you can write down the steps. Okay. So this is the resultant. This is the result of the sodium potassium pump after it has been pumped one time. So look in the 5th stage. After the phosphate is released, ATP can bind once more, so it is ready for the next cycle. Next cycle means next binding of sodium ion. This is completely based on molecular basis. So that is the reason I went for the structure of the pump. Now, how this will create an impact in the cell? Let us check how it. How it will create an impact in a cell. Just consider a cell, a normal cell containing 100 positive ion and 100 negative ion. So this I can say that this system or this cell is neutral in charge. Neutral means there is no charge left to make any polarity, right? That is the condition. So after one sodium potassium pump, what will be the condition of the cell? Look, if one sodium potassium pump is going through it, it can pump 3 sodium outside and 2 sodium inside, which means I can say that only 1 positive ion is being transported outside. Am I saying the correct thing? Yes. 3 went outside. From that 3, 2 came inside. Just consider 3 positive charge went outside, 2 positive charge came inside, because sodium and potassium exist in cationic form. So actually, in a one cycle, 1 positive ion went outside. I can say that. So after one cycle, what happens? 1 positive ion went, so inside become 90 positive ion and 90 positive ion and 100 minus ion. So now there is a slight change in the polarity. Neutrality or neutral condition now begins to change. So what happens from this condition? What happens after a 50 cycle of sodium potassium pump? What happens in a 50 cycle? 50 cycle, we can see that only 50 ions are left here and minus 100 ions are left. So now what happens? Now there is a change in the polarity. Now the cell becomes polar, which means the cell will be having more positive ions outside and less positive ions inside, which means the cell will become negative in charge. I'm talking about the plasma membrane because in the plasma membrane the pump exists. So here the chance of high positive ion and here it will be high negative ion. This is the reason for the polarity in a cell. This is also a reason for the signal transfer, conduction, because signal is electrical medium, right? So it needs an electricity to pass, it needs ion to pass the signal. This is the reason why nerve cell and muscle cell can be excited electrically, because they exist in a polar form, polarized condition. I think you got a basic idea of how sodium potassium pump helps in the polarity of a cell. This polarity is seen in a resting cell. Resting cell means a cell without any impulse conduction. Okay, without any impulse conduction. Okay, now let us see. Now we are moving to the third part, how an impulse is generated and how an impulse is conducted through a neuron. Our target is neural system. Okay, I think you got a basic idea about this. So let us move here. So let us say it once more. Look, just consider a neuron which is in a resting condition. So a neuron with a resting condition means this neuron will be having positive charge outside because it is a cell, right? It is having sodium potassium pump, and the inside the cell will be having negative charge. This is the condition of a resting cell. So the resting cell contains a potential inside the cell which is known as the resting potential of the cell. So what happens in a resting potential of a cell? In a resting potential, the cell will be having more minus inside and more positive inside. So it will be existing in a polar condition, polarized condition. Okay, talking about a nerve cell. Every cell in our body contains these type of channels or transporters or pump within them. I am just considering the neuron cell here because we are studying human neural system. So this is the condition of a resting cell and it is known as the resting potential of the cell. Resting potential of the cell is minus 70 millivolt. Minus 70 millivolt. Inside potential minus 70. Okay. Potential means potential difference outside and inside. It is actually inside it is calculated as minus 70 millivolt. It is calculated with certain experiments by using Ernst equation, by looking the concentration of sodium, potassium inside. Actually, it is done with that. Just we need to understand that the cell, when it is resting, it will be having negative charge inside and positive charge outside. Now let us move what happens if a resting cell is hit with a signal. Okay. Before saying that, you should also remember that apart from sodium potassium pump, there are certain other pumps also, right? Some may be pumps, some may be not pumps, but all these are protein molecules. I will say it once more. In the plasma membrane, there will be presence of different transporters. I'm not saying pump. Pumps also will be present. I have said the category of pumps. Apart from that, there are also protein which are not pump. Protein which are not pump means which do not require any energy, but it can transport some molecules without energy from high concentration to low concentration. The reason there is a transporter is that the molecule can't cross the cell without a transporter. One of the transporter protein is the channels, ion channels. Ion can't cross a plasma membrane because ions are usually in the cells. Ions are covered with water molecule, so they can't cross the cell. So they need a separate channel for them to cross. Channel, just consider it will be a hydrophilic tube. Hydrophilic tube means charged tube, so that any ion inside can move out, any ion outside can move inside, and any ion inside can move outside without any energy. Why I have drawn K plus and Na plus here? Because Na plus is a major extracellular ion. Without any cause of energy, it can travel inside the cell. This is a one method of transportation without any energy. Potassium is a high extracellular ion, intracellular ion. So without any use of energy, it can travel from inside to outside through the channels without any liberation of energy. Without any utilization of energy. So channels are present inside the cell with pumps also. We have, I will say an example of a pump, of a organelle lysosome. Lysosome is we have said that lysosomes are suicidal compartments of a cell, suicidal bags of a cell, because these lysosomes contain high acidic pH. Any molecule entering the lysosome will be destroyed because of the high acidic pH. How this acidic pH is maintained? It is with the help of H plus pump. Consider it as a lysosome. A lysosome will be having an H plus pump here. What does H plus pump do? It pumps H plus inside with the utilization of energy. So it is also an ATPase. As the concentration of H plus increase, you know that the more the chance the inside will be acidic in nature. Okay. So this is the condition. Now ion channels, they are channel or transport protein channels. They can be non-gated, ungated. Non-gated means certain channels are completely open. They have no any gate. They are completely open. And certain gated channels are also there, which means they have certain gates. Only in the presence of electric variation, this gate will be opened, so that ion can move outside or ion can move inside. Next, ligand gated. Only in the presence of certain ligands, the gate will be opened. In the absence of ligand, the gate will be closed. Basic understanding. Actually, this is not needed for your basic understanding only. So let us go. Now we are coming to the end of the lecture. These 2 lectures, we are coming to an end. So what happened in a resting cell? I said that in a resting cell, the inside will be negative and outside will be positive. So this is a plasma membrane. This is a plasma membrane. Inside will be negative. Look, negative charge. Okay, negative charge. And outside it will be positive. Positive means it is due to the continuous working of sodium potassium pump. Their concentration have also been given here. High concentration outside, low concentration inside sodium. It is why the triangle is inverted, because the ions will flow from outside to inside if it is a channel. Okay, so in a resting cell, this is the normal condition. Cytosol will have negative side, and the plasma membrane will have outer plasma membrane will have a positive charge. Okay, so what happens if a signal is approaching? What happens? A signal is being approaching. How signal is approached? Look, how a signal is being approached. Look, I will make a copy of it. Look, So this neuron, the first neuron, will be conducting electricity and it will be releasing neurotransmitters here. The neurotransmitters will make changes in the postsynaptic neuron. The postsynaptic neuron will have some changes. It will make certain channels open. This is the thing to be remembered. Certain channels open inside the cell. Certain channels like sodium channel. What happens in a sodium channel? Look, if it is a cell, right and left and right are having normal condition. I am saying that here a cell has been excited. What happens in an excited cell? There is an open cation channel. Example of sodium channel, not sodium potassium pump. I'm saying sodium channel gets opened because it is a voltage gated sodium channel. In the presence of voltage, this channel open. What happens if a channel is open? Especially if a sodium channel is open, sodium will flow from outside to inside. So the cell inside will be becoming positive. This is how an impulse is generated. I think it will be a little confusing for you, but I can't do anything. If you are having any doubt, please text me in your comment box, and also you can mail me. So an impulse is generated like this. Certain chemical changes occur in the cell body region, which will allow certain channels, voltage gated channel. And not voltage gated means this chemicals in the cell body will make channels open inside the axon hillock region. In the axon hillock region, before the channels were open, it was negative. But when the sodium channel got open, sodium will come inside. So the negative will become positive, and the charge will be spreading. It will be only spreading from the axon hillock region towards the axon termini. It can't go back. Actually, there is a mechanism behind it. I'm not saying because you will be getting confused. So here the positive charge will become. So what this positive charge will do? This positive charge will open the rest of the sodium gated channel. So what happens? The cell become more and more positive in condition. More and more positive. So thus the positive region will be depolarized. Depolarized means now the signal depolarized condition occurred. In your textbook, how an electrical signal is being transmitted is given in your figure 18.2. Just look here. In this area the cell is in resting condition, this area, which means positive outside and negative inside. But when a stimulus or when an action potential is being hit in this area, what happens? Certain channels get opened. These channels will allow the certain channels means sodium channel will open. So sodium channel will make the sodium outside to come inside, because sodium is extracellular ion. So it will become a positive charge here. Now the positive charge will make this voltage gated sodium to open. So sodium gets inside. So here the sodium gets accumulated and the signal is being transmitted to here. So what happens? Here positive charge increases. This positive charge will make the adjacent sodium channel to be opened. So here the positive ions get increased, and this is how an impulse is being transmitted from one area to another area. It can't go back. I'm saying it can't go back. It only go back to one direction. It is because there is a mechanism in the voltage gated sodium channel which will allow the movement of the signal only towards the axon terminus, because the sodium channel is having a mechanism behind it. If I am saying this, it will become a lengthy lecture. That is the reason I am not saying it. Actually, it is not needed for your NEET purpose. Because it is a conceptual topic, I am taking it. I am not going much deep inside it. Okay. This is how a nerve impulse is being generated and it is being transmitted. So if we go back, okay, look. So this positive charge will make open. So open this voltage gated channel, so it will make sodium outside to get inside. So it will become positive. This positive will make the next open, because before it were all closed in the resting condition. This is how an impulse is generated, an impulse is being transmitted. Actually, this was so an action potential will make certain channels open here. It will allow the opening of the rest of the channels here. You should understand that the channels are only present sorry, channels means the sodium potassium pump or any channels are only present at the node of Ranvier. Node of Ranvier is only present in the node of Ranvier. It is not present here. Which means, look, if it is a myelin sheath, it is a myelin sheath, and it is an axon. It is, let us take this as an it as a node of Ranvier. That only in the node of Ranvier the channels and pumps are present. This is to be noted very importantly. In the myelin sheath, they are absent. Okay. So we can say that an impulse will be generated here. Okay, this will allow, in a resting condition, the cell will be having negative charge inside. So actually a positive charge here will make these all negative. And here a pump opens. Pump means a channel opens. Here also channel opens. Here also channel opens. Here also channel opens. If the axon is very much in length, all the channel get open in the Ranvier region, node region. This is why we can say the action potential is jumping in nature. Okay, this is the reason. So one thing to remember. So actually this is the end of the lecture. I think if you are having any sort of doubt, you can text me in the comment box. I will be going through the comment box. Okay. So there is also one thing to be remembered. The neurotransmitter, which I have mentioned in the earlier part of the lecture, is of two types: inhibitory and also excitatory, which means certain transmitters, when released, have excitatory nature, which means when these are released, when these get bind towards the next postsynaptic neuron, it will excite the neuron, which means it will make open of the channels and it will make positive insight. So the action potential is being passed towards the next neuron. But certain neurotransmitters are inhibitory in nature, which means these will be released, neurotransmitters will be released from the synaptic knob towards the synapse. The synapse, after, they will get bind to the dendrite region, but in the dendrite region, in the axon hillock region or the cell body region, they will not open the positive charged ion, but they will open negative charged ion. So the charge inside the axon hillock will become more negative. So there will be no action potential. So for this reason, they are known as inhibitory, which means they can't pass any action potential to the next neuron. So actually, this is the end of the small topic, conceptual topic. If we get time, we can go through the previous year questions also. Okay, I hope you understood it. Everything is given in your textbook, the definition, everything. Okay, thank you. If you are having any sort of doubt, you should text me in your comment box. I hope you like my video. Subscribe it and mention your valuable comments. Okay, thank you.